Hello, kiddos. Welcome to biology. We're going to be talking about gene expression today, which is simply the use of DNA to actually express a trait by building a protein. So you can see in this slide that there is a tobacco plant expressing a firefly gene. When we say expressing, we say that it's producing the protein to glow. So they inserted the gene into the DNA of this tobacco plant to create a protein that glows, and now the tobacco plant can glow in the dark. The same thing with the jellyfish gene that helps jellyfish glow, and they inserted it in pigs, and now you've got glow-in-the-dark pigs. So if I was to look at RNA versus DNA, we're going to be talking about two different types of nucleic acids in this unit. One is RNA, ribonucleic acid, and one is DNA, which we've been talking about for ages now, deoxyribonucleic acid. So in both of these, they're both nucleic acids, so they're both made out of strings of nucleotides that have a 5-carbon sugar, a phosphate group, and a nitrogen base. There are three big important differences, though, between RNA and DNA. First of all, the sugar in RNA is ribose. In DNA, it's deoxyribose. In RNA, it's a single strand. In DNA, it's a double-stranded helix. The bases in RNA are adenine pairs with uracil. So this is different. Adenine pairs with uracil and guanine still pairs with cytosine. But in DNA, thymine and adenine pair together, and guanine and cytosine pair together. So we've already done this. Now we're going to be talking about, well, how do we take this and create a protein from it? And that is going to require RNA. So I think of DNA as like this giant master copy for you. If you were like a builder and you were on a construction site, they would have a master plan that has all of the information needed to construct an entire building. But builders don't bring that valuable master plan to the construction site, okay? It could be damaged, it could be lost. Instead, they keep that master plan inside of the headquarters. And they just copy off parts of the plan and give those to the contractors that need them. For instance, electrical, they just get a plan of the electrical work. Plumbing just gets a plan of the plumbing work that needs to be put in. Okay, and they take that smaller piece of information that's just necessary for that builder at that moment, and that copy is what goes to the build site, and then the builder can use that to build that section of the building. Okay, so DNA is kind of like this big master copy that has all of the instructions to make you, and then RNA is going to play three very important roles. First, it's going to be like that copy of the blueprint that has just a piece of the total master plan. Second, it's going to act as the builder, which is going to actually piece by piece build together the new protein that's being expressed. Then the third is it's also going to act as the supplier. So it's going to go get the supplies and it's going to bring it. Okay? Now I'm talking about three different types of RNA. Here is a picture that shows those three different types of RNA. You've got mRNA, called messenger RNA, which acts as a copy of the DNA, a copy of that master plan. The mRNA, as the messenger, can go between the DNA, which is protected in the nucleus, all the way out to the building sites, which are in the rest of the cell. The building sites are called ribosomes. Remember, we know that ribosomes are a non-membrane bound organelle. So they don't have a membrane, but they are considered to be an organelle, and they make proteins. Well, ribosomes are made out of ribosomal RNA. So these builders, these factories for protein, these guys are actually made out of a type of RNA called rRNA. tRNA, or transfer RNA, transfers the amino acids, which are just in that cell soup floating in the cytoplasm. They go out into the cytoplasm, they pick up an amino acid, and they bring it back to the building site at the ribosome. If I was to look at this in a diagram, it would look something like this. Here's my cell. This whole thing is the cell. This right here is a nucleus, and this area represents the cytoplasm. So you've got your DNA protected inside of the nucleus. It can't leave. It has to stay in the nucleus. So what that does is it opens up, it transcribes a copy of the DNA into an mRNA molecule. Then that message is edited. We call that RNA processing. It's edited and it's packaged up with a little cap and a little tail that's going to protect it as it moves out into the cytoplasm. It's going to swim out into the cytoplasm. It's going to get picked up by a ribosome. The ribosome is going to read the message as it slides through. The tRNA is going to pick up amino acids and bring them to the factory and that polypeptide, which, as you know, is the polymer of proteins. That polypeptide is going to be produced. So we say there are three major parts to this process. There's transcription, making of the copy, editing the copy, RNA processing, and translation, when that copy of the message gets actually turned into a polypeptide or a protein. This represents another model of that process. Here you can see the DNA molecule, and here this color represents this very specific gene for a specific protein. It's going to open up, a copy is going to be made, a complementary copy is going to be made of that gene of interest. Then that message can be read and converted into an amino acid sequence. That chain of amino acids is called a polypeptide. Transcription is making the copy. Translation is turning that message into an actual protein. But it's actually not a protein yet. So you can see that this right here represents an amino acid sequence. It's not a protein until it's folded into its three-dimensional shape. So we call this its primary structure. Its secondary structure is when it gets turned into the sheets or helices or rods, and then those helices, and rods are then refolded into a three-dimensional shape, which is going to be a functional protein. Now, sometimes proteins are really complicated and they require more than one protein working together to get the final result. Here's an example of a protein that is just in one protein working by itself. Insulin is a protein. Here you can see the amino acid sequence. It all gets folded up into this molecular shape. Kind of looks like a clump to us, but if we were to use computer digitaling to break it into rods and helices, it would look something like this. I'm not going to explain to you how it works. It's complicated, but this is just a simple protein in its tertiary shape. This, however, is a protein that requires more than one protein to work together because it does a more complicated job. You can see right here in this video that this is a moving, functioning protein. This is kinesin. It's a motor protein that moves things around inside of your cell, okay? This, you can see this is one protein. There are two proteins working together. This is another protein here. Here's a protein, and here's a protein. So this, we would say, has a quadentary shape. So it has its primary amino acid sequence. It got turned into rods and helices. Then it got folded together, right? That's a tertiary structure. And then several tertiary structures were put together to make this fully functioning machine. All right, kiddos, I know this is really complicated. So what is it that you actually need to know? First of all, you need to know what happens where. So we need to know that transcription happens inside of the nucleus and that turns DNA into a complementary RNA strand. But it's not ready to leave the nucleus yet. It has to be processed. So the RNA is processed, once again, inside of the nucleus. You have to know that DNA must stay in the nucleus where it is protected at all times. We are not going to let that master copy of you be exposed to anything in the cytoplasm. Little tiny pieces of that master copy can exit and go to the ribosome, which is the protein factory. tRNA is also going to be in the cytoplasm, so we're in the cytoplasm now. Translation happens in the cytoplasm. That tRNA is going to go out into the cytoplasm, pick up amino acids, and bring them in. And the RNA is actually going to put them together into that polypeptide. After the polypeptide is put together, it can be folded into the full functioning protein. Okay, kiddos, so let's take a look at this molecule. This is an RNA molecule. How did I know? First of all, doesn't have two strands. DNA is two-stranded, double helix. So it's this single-stranded. In addition to that, I have another important clue. It says AUG. AUG. It is using uracil to pair with A instead of thymine. So if I had the original DNA strand here, the DNA strand would be TAC, right? Because it would have thymine. But the A from the DNA molecule is going to pair with U in the RNA molecule. Does that make sense? Okay, please note that these have been broken into three little segments. Okay, so 3 nitrogen bases form one codon. A codon is what codes for a specific amino acid. So it takes 3 nitrogen bases to code for one amino acid. So let's take a look at a chart that we could use to figure out what AUG would code for. We use an amino acid chart that looks like this. You can see that you start over on the left-hand side, A, then you have U, and then you come across G. Well, AUG is methionine, abbreviated as MET, or the start codon. Here is another type of chart that I like even better. You start here, A in the middle, U, and then G right here is methionine. Once again, the start codon. Let's take a look at the second one, AAC. Using this chart, I would do A, so I'm down in here, and then I go across to C. Oh, ASN is my abbreviation. On this one, AAC, asparagine. Looking at this diagram, you can see that this is just one more model of transcription and translation, or gene expression. Here you can see the DNA is being unwound, a copy of mRNA is being made, the RNA is processed, it leaves, it goes to the ribosome, where the ribosome reads each individual codon to know which amino acid needs to go in order. What's going to bring those amino acids to the ribosome? The tRNA. Take a moment to watch this translation animation. Okay, let's do the protein synthesis worksheet. First of all, we're going to start off with DNA. You know how to do those base pairing rules. A pairs to T, T pairs to A, G pairs to C, G pairs to C, T pairs to A, etc. I'm going to let you finish that. Next, we're going to transcribe this bottom strand of DNA. So where there was a T, there's going to be an A. Where this A would have paired to T in DNA, instead in RNA, it's going to pair to U. So this should be AUG. Okay? AUG. And then keep going, and remember, instead of thymine, you're going to use uracil. Next, you're going to read them 3 at a time. So AUG is your first 3 right here. AUG is your first 3 right here. And you're going to then use this diagram in order to read that code and turn it into an amino acid. So remember, AUG is methionine. That is the start codon. We're going to abbreviate these with the 3 letters, which is why I have this diagram here as well. So methionine is written over here, so you know what that stands for. And MET is here so that you know how to abbreviate it. We're going to go ahead and put in MET here. Next, we've got GUA. GUA, right? So if I was to read GUA, then I would be looking at GUA, that's valine, VAL, valine. Then you are going to do the next three. The next three here would be GCT, not TU, GCU, because we're in mRNA. So then we go to our chart. GCU is alanine, ALA. GCU is alanine. Okay, so we're going to go ahead and we're going to continue to code for those amino acids. So fill in the rest of the amino acids. Remember, mRNA is synthesized in transcription. mRNA has codons, or sets of 3. tRNA actually has the complementary anticodon. We're not really going to talk about that. I just need you to know that the complement to this is how it knows it has the right amino acid. So we read the message, we read the mRNA, and we use the mRNA in order to determine the amino acid sequence. Let's see if you can do the next one on your own. I'm going to let you finish out this worksheet, and I hope you do a great job. Good luck.